Thank you so much. Very nice information. Thanks a lot for very nice talk. Thank you. So I will starting asking first, and later we will have more questions to come. So can you come back into the last slide?
Okay, sure. Last slide. Oh, I need to share my screen.
Yes, please.
Last slide is oh, it didn't go back to the last one. This
Yes,
slide?
yes, this one. Yeah. So the pipeline for the laboratory on the left hand side, on the at the bottom of the left hand side, we can see that for the microbiome detection, it's around 6.5 hours. And then after we identify which microbiome, and then we detect the AMR 2 more hours. So in total, we have around 8, 8.5 hours, isn't it?
Yes. I think this is a timeline that
Mm-hmm.
in average
Mm-hmm.
from the you get this sampling and the median turnaround time is within 6.7 hour you're able to detect the microbial.
Mm-hmm.
With sometimes you already have the enough data for MR detection, but sometimes you may need a little longer time for a more precise MR detection. And
Okay.
by 16 hours, you get the whole genome being assembling. So you get all this information before your standard, 40 hours cultures data come back.
Mm-hmm.
Yeah.
Yeah. So and then my question is, um, this one, do we apply the adaptive sampling into it? Like we give a set of the AMR gene database um, for the software in order to do something like targeting them
No,
or
no, no. This is a metagenomics, so they don't do any adaptive sampling.
Mhm.
So they will find if anything that's inside there,
Okay. So
regardless of
very
whether it's bacteria or fungi or DNA or RNA viruses.
Okay. So I just think that they will do some type of adaptive sampling that give a list of on the AMR gene into
Mm.
the software. And then if they detect them, and then they will sequencing deeper for the
Mm.
gene, otherwise they don't. So it
Okay.
not so we sequencing everything.
Yes, in this case, they're sequencing everything.
Okay, perfect. Yeah. So and then we have some question in the chat box. The first one is how the real-time error correction work, especially give the high error rates associated with the nanopore data.
Let me see the chat.
Yes, it's very beginning of your slide. So you
Uh,
mentioned something about the
sure,
error
sure.
correction. Yeah, really.
Okay. All right. To answer that questions, right? Indeed, if let's say it's a real-time analysis, right? Because we have different workflow that you can select or different tool that you can select. With the real-time analysis, of course, the accuracy you may have a higher rates because the accuracy is lower. But it allows you to sort of like knowing like whether you have already have sufficient data to stop the sequencing run already. And you can actually then do another analysis with a more is with the better or finer workflow that give you a more accurate or higher accuracy data sets. And I think when it comes to because if you are familiar with nanopore sequencing, you'll know that there are three type of base calling model. Fast. This one will use the least computing power. It will enable you to get the sequence of fastest. However, it also has a higher error rates. And then the second mode is high accuracy model. The third one is sub super accuracy model. But however, because the bacteria genomes are typically very small, so there's between fast, hack, and sub, there's not much differences in terms of very, very significant in terms of the computing capacity required for between hack and sub best calling model.
Mm-hmm, mm-hmm.
Mm.
Okay. So and then the next question is about what sample preparation consider are essential for detecting the AMR gene effectively with the nanopore sequencing, especially in the complex microbiome.
I think it is for microbiomes, I think it depends on the sample types, like whether it is from soil, is it from stool, or from different types of microbiome. I think one of the challenge remain on extracting high quality um, genetic materials from all these
Mhm,
samples. So you
mhm,
may need to try different extraction protocol. So in Nanopore workflow, sorry, in Oxford Nanopore, right, we actually in our website, there's one sections that has extraction protocol. You can actually search depending on the sample types that you are working on. You can try to search whether or not there's a collection or the past work that have been done,
mhm,
and then it's showing good result with the Nanopore platform, then you can actually try that particular extraction methods to ensure that you get good the good material to start with.
mhm.
And
How about the blood
providing,
sample?
hmm? Sorry,
The blood one, the raw paper, blood,
blood. Yeah,
yes.
blood is very common. It is it's very, very common. We do have that protocol in our website.
Okay, okay, thank you. And the last question from Huy, he asking, can ONT real-time analyst um, capa- capability to be applied to the AMR detection for rapid point of care testing? If so, what are the main limitations?
Mm. I think definitely yes. I think, but of course, it depends on your definition of rapid point of care testing. Are you saying 45 minutes or 30 minutes or when 1 hour?
Yeah.
But no matter how we have use cases of like adaptive samplings that are able to diagnose a newborn within 3 hours of birth.
Mm-hmm.
So I would say that it is possible, like for example, using adaptive sampling. As you mentioned, Dr. Loy, that you have this idea of detecting the MR with adaptive samplings. I think that can be developed definitely. And the main limitation is that with adaptive samplings, you do need to ensure that the computer that you have has the enough or the sufficient computing power to catch up with the adaptive sampling, because it does require a little bit more of computing required than the normal sequencing.
Yeah. Yeah.
Mm.
And I just want to asking about the CIVD in the because we want to apply in the hospital
Yeah.
about healthcare, and
Mm-hmm.
then we have the any available solution for the CIVD.
Mm. Okay. Our future path, for Nanopore is definitely one of the focus is on the clinical application. So a lot of customer using us for clinical LDT. However, for IVD, we do have our devices, Grid Ion, that are going to have Q lines. And after the Q line approval, we are also going to submit for IVDR. So in terms of the IVD kit, we are partnering with bioMaru. So we will have one ample TB kits that are going to be launched with bioMaru very soon, maybe end
Mhm.
of this year or early next year. But please stay tuned with the release of our first IVD kits. And with that, there are also some applications that we are developing to add in, add more and more menu under the, for diagnostic use
Yeah,
after the TB. Yes.
yeah, yeah. Thank you a lot. Are there any more questions, please? It's very nice talk from Tan. Very nice.
I
So,
have one question for the wet lab protocol.
Yeah,
Uh,
sure. Go ahead.
in the QC step, if I use some kind of kit for site selection, then should I use uh, uh, staff station or bioanalyzer to QC the sample again?
Um, if I understand you correctly, your question is asking whether to use bioanalyzer or tap station for QC, right?
Yes.
Oh, it's fine. Depending on your labs, whether bioanalyzer, you have bioanalyzer or you have tap station, both are fine.
I mean that if I use the protocols, if I use some kind of kit like some kind of kit that used for selection of the fragment DNA, then should I use uh staff staff station for quizzing my sample again?
Yeah, sure. I think it's optional, but you can definitely can because it will give you the you will visualize, how effective is your size selection. You can see whether or not there's, any smaller fragments in or is the profile what you are expecting. It's optional, but it's good to have.
Uh, uh, there are any requirement for deny for distributor of deny fragment when I input the sample. I mean that I gonna sequence whole genome sequencing. Uh, then should I then do there are there any re requirement for distribution of deny fragment?
Do you specifically refer to this one? For this one, the NOMIS protocol? Sorry, you're really referring to this one?
Yeah,
Oh,
about that
it's not. Yeah. Okay. Yeah. Okay. It's not required. So it's not compulsory for you to um um really need to do the fragment analysis before you start sequencing. It's optional.
yeah, it's optional, so you don't have to.
Yeah,
Thank you.
you don't have to.
So
yeah,
next we have Paul. Are you okay with the answer or you want to asking further?
it's okay.
Okay, thank you. So we have one question from you. Am motor enzyme rapidly sequencing adapters? So I even don't understand the question. So can you please elaborate your question, please?
So in I see in the slides, the purple color mean the rabbit sequencing adapters. Is that motor enzyme?
Oh, okay. So that motor enzyme, right, is important because it will help to control, the movement of the DNA or RNA when they go through the pore.
So
You're asking that the motor enzyme in the sequencing adapter?
yeah.
Yes,
Um
they are together. The
so
rapid
the mo
sequencing adapter.
yes, is that the same enzyme?
Same enzyme.
So on on the right on the right side, the purple color. I mean, it does the motor enzyme you mm,
Uh,
on the
uh, Excuse me,
on
maybe
the
I can help to address this question. So allow me to explain it in Vietnamese for Miss Dương. Chị Dương ơi, câu hỏi của chị là cái phần motor enzyme và phần sequencing adapter ở trong biểu đồ đúng không ạ? Thì
đúng
ở
rồi.
vâng, thì ở trong biểu đồ thì mỗi cái sợi DNA hoặc là RNA sẽ được gắn một cái phần adapter, và motor enzyme là cái cục màu tím thì nó sẽ bám vào cái phần adapter đấy. Ờ,
Ở đây, cái cái motor enzyme đó là nó nằm trong cái kit chuẩn bị thư viện của mình đúng không?
Đúng
Hay
ạ.
là nó nằm trong cái flow cell,
Nó là một thành phần trong kit chuẩn bị thư viện ạ.
là nó gắn vào adapter?
Vâng ạ.
Ok, cảm ơn em.
Vâng ạ. Vâng.
Okay, thank you. Thank you, you. So, please, if you have further question, you can send an email to BCE over here. We have Thanh Dương, who is the representative for BCE in Vietnam. Yeah. So, mọi người mà có thêm câu hỏi gì thì ở đây có bạn Dương, bạn Thanh Dương á, là đã gỡ cái contact ha. Thì BCE thì là cái nhà phân phối Oxford Nanopore của Việt Nam mình ha. Thì mọi anh chị em đây có câu hỏi thì cứ thoải mái ha, gửi email đó contact ờ, cho bạn Thanh Dương. Ở đây nè có địa chỉ email rồi mọi người. Rồi, ở đây còn có ai có câu hỏi thêm nữa mọi người? Are there any more question please? Các bạn có thể hỏi tiếng Việt rồi xong xuôi đây có Dương nè, rồi có tụi tôi sẽ giúp dịch sang tiếng Anh. Mọi người thoải mái là mọi người. Ở đây là cơ hội của mình, không tốn tiền, không gì hết, không hỏi. Chi vậy, đúng không? Đó mình nghe, mình đã dành tới thời gian này rồi thì mình phải tận dụng vào. Đó người ta nói một cái bài quá trời hay luôn, quá trời thông tin luôn. Không biết cái gì thì hỏi đi, chứ mốt xong rồi tan rồi thì hỏi đâu có ai nhớ, đúng không? Đó, hỏi đi mọi người, không hỏi thì tan á. Tôi cũng buồn ngủ lắm rồi, nhưng mà tôi cũng muốn đã làm thì làm cho nó tới tới nơi tới chốn chứ. Có ai hỏi nữa không? Rồi không ai hỏi nữa thì thôi nha, tôi kết thúc đó, đừng có hối hận nha. So
Ờ, cho Dương hỏi một xíu là cái slide cuối mà bác Mavis Tân trình bày á, nó khá là hay, nhưng mà không biết là áp dụng ở ở các bệnh viện ở Việt Nam mình á, thì chi phí cho một bệnh nhân như vậy á thì nó có khả thi không? Cái này không biết hỏi ai.
để mình hỏi. Mavis, please
Yes.
re-share your slide. So she asking that the
Oh,
idea of the method using the Oxford method to reduce the time is very good. So she just want to asking that it's possible to apply in the clinical setup in Vietnam. She want to know the code. How
which one? Which slide?
the last line?
Oh, the last slide.
Yes. That's why I
Okay.
asking, yeah, about the using the orthogonal to discovery the detection the bacteria as well as the AMR for lungs uh, infection disease.
Yes. So for implementation of the, in fact, this particular um, study
Yeah, huh?
is in ICU settings, is clinical.
Where
Um,
do you know where it happened?
it is in the Guy's and St. Thomas in the UK.
Okay.
Yeah, so one of the PI, his name is Professor Jonathan Exworth. So
Mm-hmm.
he's one of the lead PI for this program. He is also currently working with Nanopore part-time as a medical affairs.
Mm.
If you are interested to bring in to your hospital, I'm very happy to connect you with Professor Jonathan so that you can, discuss this in further details
Okay.
on, how can you actually bring this as well. And in fact, um, he's here in Asia a few months ago, and he's going to be here in Asia as well. And but maybe this trip, he will not be covering Vietnam, but the subsequent trip, he can actually visit Vietnam and to discuss this in further detail with you as well.
Thank you. So because we don't see any publication, so we rather to read the publication first and then
Oh, there is a few. I share with you,
okay,
and you can share with the rest of the group.
okay,
And like for example, this one just got published in 2024. And please feel free to, read it because this is a very detailed articles.
Mm,
And if you go to the supplementary, the data, they also describe in details on, how they actually do this. It's very, very
okay.
good reading. Yeah, please,
Mm,
I'll share this with you.
okay.
It will
Thank you.
describe in details what have been done.
Yeah, yeah. So I refer to analyze the data to see how it look like first, and then we will thinking about asking further information. Yeah.
Yeah,
So
sure.
because the
Feel free to
brand,
reach out to us, or BCE, and then they will reach out to us, and we can always meet.
yeah, yeah. Mm.
Can I please have one question?
Please go ahead.
I have the my min ion, and then it says that it's just the min ion is one flow cell, and then the maximum uh, uh, of the sample we can run on the min ion for one flow cell. Is it depending on the kit I use, like 24, the kit with 24 barcode or kit with 96 barcode? So what's the maximum sample I can run in at one time?
Um, I can you repeat your question again? What I get is that how
So
many samples can be run in one
yes,
floor cell, right? For which
yes.
application?
Um, for whole genome sequencing, like to close genome.
Oh, okay. So for whole genome sequencing of bacterial isolates, the NOMIS protocol that we have optimized end to end, that one allows up to 24 samples to be multiplexed in one floor cell.
In one flow cell.
However, if you have
Um,
less than
in
that, let's say you only have a few samples, right? You
yeah,
don't need to run it for a full 72 hours. You can actually run it shorter, and then once you generate, you get the enough data, you can actually stop the sequencing run and then use the floor cell for next run. But if you have a full 24 sample, you can let it run for a full 72 hours, and then at the end of the experiments, you will get all the data that you need. So with Nanopore, you always have this flexibility. So it will be very subjective to answer, how many samples to be multiplexed in a run, because it depends
mm,
on the number of sample that you have, the coverage depth that you require, and the application as well.
yeah, yeah. Thank you.
Mm-mm. In terms of barcodes, you can multiplex 24 or 96, and even there's a publication that published last year in 2023. It is a article that is describing how they actually barcodes 100K specimens in one single nanopore runs. So do like a dual index kind of things to increase that barcoding multiplexing capability. Yeah, so a lot of things can be done on this platform.
Yeah, thank you.
Yeah, you're welcome.
Thank you. So any further question, please? Okay. If there is no more questions, please. We have the link for register for next lecture. Thank you. Thank you again for Mavis and as well as Ray and Ting. And also thank you for BCE and BKSH that helped us to invite very good speaker. If you have any further question, please send the email to the BCE or the KSH. They will transfer the information to the speaker today. So
Yes.
thank you again for a very nice talk, very nice information. Yeah.
Thank you, Dr. Louis. Thank you so much for everyone who joined. Thank you.
Thank
Thank you,
you.
thầy Lợi. Thank you, everyone.
See you next time. Yeah.
Thank
See
you.
you.
See
Bye
Thank
you.
bye.
you.
Um,
Thank you.